The DNA replication process is semi-conservative, meaning it keeps one parent strand in each daughter double helix. To initiate replication, helicase, which is right here, binds to an origin of replication and then continues moving down the DNA to unwind and separate the DNA strands. Here we show the helicase already starting to unwind the left strand. In the front, topoisomerase prevents the helicase from overwinding the DNA by cutting the DNA and letting the pressure go.
The single-stranded binding proteins hold the strands apart, preventing them from reannealing. And then the primase, which is shown in red here, creates the RNA primer, which begins the process of extending the DNA, which is the job of the DNA polymerase III. The DNA polymerase III extends the DNA from the 5' to the 3' direction from the primer. The leading strand has a continuous movement, while the lagging strand forms Okazaki fragments that are discontinuous.
In the lagging strand, DNA polymerase I removes the primers and replaces them with DNA. Then, since it cannot connect the DNA fragments, DNA ligase must connect them. The DNA replication process does have an occasional error, which is fixed by nuclease through cutting the wrong DNA, DNA polymerase III replacing it, and ligase attaching them together. In eukaryotic cells, the fact that the DNA polymerase can only add nucleotide bases to the 3' ends of the primers makes it so that the ends of the DNA cannot be replicated. However, there are repeated patterns of telomeres that are lost over time, which are maintained by telomerase. These postpone the loss of genetic information over time.
This has been DNA replication. Bye.